Okay, so good afternoon, everyone, and thank you for joining us today. Today's hosts are the American Brain Coalition, the American College of Neuropsychopharmacology, and the Co-Inventorins Bioscience. And we're honored to be having a special discussion with a leader from the Drug Enforcement Administration, who will talk about the barriers and challenges to research with psychedelic compounds and explain the issues surrounding scheduling. So I'm Mark Resnick, and I'm the American Brain Coalition's incoming chair, as well as a fellow of the ACNP. And I'm also a professor of physiology, biophysics, and psychiatry, and director of the Biomedical Neuroscience Training Program at the University of Illinois College of Medicine in Chicago. And I'm also a research career scientist at the Jesse Brown VA Medical Center, also in Chicago. A representative from FDA was to join us today, but recently we learned that they're no longer able. However, we're thrilled to have a few special guests from the DEA here to provide a short presentation about Schedule I registration process and answer related questions. But before we hear from our DEA colleagues, I'm honored to introduce Tristan Wimmer, a former Marine Corps sniper, to tell us his story. Tristan?
Tristan, you're on mute.
Yeah, always one. Yeah, thanks for having me. Thanks for the introduction, Doctor, and thanks for the invitation to speak. My name is Tristan Wimmer, and I am the founder and CEO of 22 Jumps. 22 Jumps is a fundraising organization that uses base jumping as a platform to support the development of traumatic brain injury testing and therapeutics. The organization was started 3 years ago in honor of my late brother, Kieran Wimmer. He was also a Marine. He was a recon Marine and a MARSOC Marine, and we were in 1st Marine Division together at the same time. And in 2006, he suffered a massive traumatic brain injury in Iraq, and from the backblast of basically caught the entire backblast of a law rocket to the face. Injuries were extensive, brain swelling, facial bone fractures, secondary fragmentation, burns, busted tympanic membranes, like the whole thing. Very, very long recovery. He ended up staying in to redeploy when MARSOC was stood up, but we noticed pretty quickly thereafter he was having some issues, and he basically the issues got worse and worse and worse, and there was a lot of volatility in his life, and is unable to sort of manage a lot of aspects of his life. And so about 9 years later after his injury, he committed suicide, took his own life in 2015. I was actually working overseas for the State Department at the time when I got the call, and it was devastating. My family and I were pretty devastated, and so I was looking for a way to honor him. It took me a couple of years to get my feet underneath me, but I was looking for a way to sort of fundraise in his name. And before he had died, he actually inspired me to get into parachute sports. I had found climbing right after the Marines, and then he found parachute sports, and so I followed him into parachute sports, and then so I wanted to. That was my inspiration to use it as a fundraising platform, and it was supposed to be a one-time event. Wasn't really looking to start a nonprofit, but we ended up raising way more money than we anticipated, and so we did it again the next year and had the same effect. And so we kind of decided to add some structure to it and actually make it a going concern. Make it a real nonprofit and have accounts and things like that. And so, but while that was very fulfilling and cathartic, I was still very much struggling personally. And I really felt like it doesn't matter how much good I tried or how big I got 22 jumps or how, I just felt like there was like the way that I describe it was like a nuclear core of like guilt and sadness that I couldn't touch. I couldn't do anything about it. It was always there. It didn't really matter what I did, and a really good friend of mine that I was contracting with, and psychedelics was not something that I was seeking out as a therapeutic. It's not really, I worked for a Fortune 100 and I got my MBA, and I was going down a path where psychedelics really weren't in my life, in my peripheral. So, but some guys that know me really well, they suggested that I go on this retreat with them, and so ayahuasca kind of found me, and I, I went on a retreat. It was a wonderful experience, and I didn't realize how much of a positive impact it had on me personally, until I got back to the States, and a lot of what I had been dealing with, sort of fixating on my brother being gone, the negative thought loops, anxiety, depression, like that kind of all dropped off a cliff, and it was much more easy to be around, much more just a lot. I was a better friend, I was a better husband, and I'm just kind of a better able to like manage that grief. I mean, it's not gone. I don't think there's anything that will make it go away, but it's much more easy to manage nowadays. So yeah, big advocate for the work that's being done and sort of what's being talked about today. So yeah, thanks for the opportunity.
And thank you. Oh, and thank you, Tristan. Now I want to turn it over to Chantel Ferlin-Beckham, who is the Director of External Affairs at Cohen Veterans Bioscience and an ABC board member, along with Leslie Ritter, the Associate Vice President of Federal Government Relations at the National Multiple Sclerosis Society and the ABC American Brain Coalition Advocacy Committee Chair. And they're going to moderate our panel discussion. Chantel, Leslie?
Thank you, Dr. Resnick. I thought I would take a couple minutes just in light of what Tristan said and thinking about the panel that we have coming up and who we're going to talk to just think about the problem. And, according to some recent SAMHSA data, over 40% of Americans have a mental health or substance use disorder. And for many of them, the traditional or existing treatments are just not sufficient to be able to treat or sufficiently treat what they're going through. And many people show resistance to those treatments. Psychedelics have kind of emerged recently as having the potential to address conditions such as PTSD, depression, substance use, anxiety, chronic pain, and even traumatic brain injury, among many other conditions. I think everybody is sort of aware of the fact that there's no shortage of media articles looking at this renaissance of psychedelics across the board. And there's even a resurgence in the amount of research. So after kind of dropping off the board in the early 60s, we're now starting to see investment into this research across the board. We're seeing the number of publications dramatically increase. And we're seeing the number of clinical trials really take flight, particularly in the last few years. But although there is this resurgence of research, as of July of 2021, published studies of classical psychedelics had only a total of 315 participants, which increased last year with the publication of 2 pivotal studies enrolling an additional 320 participants. And so more research is really needed, and that's kind of what we're here talking about today. The research that is available does show great promise. The VA recently had an evidence brief where they stated that MDMA-assisted psychotherapy for PTSD may reduce PTSD symptom severity and lead to disease remission in a proportion of cases. Psilocybin-assisted psychotherapy and ayahuasca for depression and psilocybin for alcohol use disorder may also be effective. But because of the limited amount of data and the fact that most of these trials are very small sample sizes and just limited in number of trials, there's a lot of critical research gaps that we need to address. So we need to really be able to expand this research. The psychedelics drug development pipeline is also really taken quite off. Over 45 companies have some sort of compound in development, many of these in phase 3, phase 2, and phase 1. And so there's a lot of interest in being able to expand the research in the industry side of things as well. So now I'm going to turn it over to Leslie, who's going to introduce our panel, and then we'll have a Q&A session have the DEA speak, followed by a Q&A session.
Thanks, Chantel. So now I'm going to introduce our special DEA guest today. So joining us this afternoon is William J. Hewitt. He is a doctor. He is the unit's chief Schedule I researcher and international control unit in the drug and chemical evaluation section of the Diversion Control Division at the DEA. That is a fantastic title, by the way. Along with his colleague, Dr. Teresa Carbonaro, who's a pharmacologist in the Drug and Chemical Evaluation section in the Diversion Control Division at the DEA. So Dr. Hewitt, without further ado, I'll turn it over to you.
Sure. And can you hear me just to make sure?
We can.
Okay, great. Yeah, we don't choose the titles. And just for clarification, my doctor title, I have a PhD in applied mathematics, so I'm not a medical doctor of any kind, but certainly I've done my specialty, my area is mathematical biology. I've been involved in human subjects research in the past. I was an academic. I served for many years on an institutional review board and chaired that board for several years as well. Dr. Carbonaro is with me. She's actually in Croatia right now, so she's joining us from very far away. But I'll let her speak about her research maybe when we do the panel session part. But she's in the past been very involved in research with psychedelics. And so I thought it would be great that she could join us today. And I really appreciate her joining us from so far away. So I just wanted to give a brief overview here of our Schedule I research program. Again, it's in this very long named section, Drug and Chemical Evaluation section. But we have a team of scientists in that section and working on the Schedule I research program. And our goal is really to be available to the research community to help everyone comply with regulations. The idea is to have scientific staff available to make that contact at headquarters with anybody working in the research community. In addition to, then there's somebody in the field, a diversion investigator, who would be in touch and who would be visiting in person, wherever the site may be, for that research. And our goal for today, and in reaching out and having these opportunities to speak to people in the research community, is to help eliminate perceptions of barriers. We don't want everyone to think that there are barriers preventing them from doing their research. Rather, it's regulations that we just need to comply with, and they're rather straightforward, we believe, and we can help you through them. And we want to help you conduct that research because we believe that the research is very important. It informs policy and regulation decisions, in particular for this group and the discussion today, the relevance to drug approval process. So we realize the importance of all the work that you all do, and we want to help support that as much as we can. And so thank you very much for the opportunity to speak today and giving us this connection. I will have, it was on that first page, but it'll also be on the last slide, an email contact if you want to reach out to us at any time. If you're thinking about research or have questions about it, please feel free to reach out to us. So Schedule I research activities cover everything from clinical studies, phase 1, 2, 3, through you have the preclinical studies all the way down to, say, assay development or analytical method development. So all research areas involving Schedule I substances would come to us, and there would be some sort of an application put together that the details are what I'm going to try to convey to you today. If you're looking for the information or interested in registering, you go to the Diversion Control website, and I've listed it here. And you see right there on the left side of the screen, you see a registration tab. That would take you to a place where it has a whole bunch of forms listed. And in particular, if you're a new researcher, you'd be filling out something called a Form 225. If you are an existing Schedule I researcher and you are renewing your registration, then you do a 225A. But those forms are there. They have information provided along with them in terms of how to fill it out. And again, we are always available to help you fill it out. Not as obvious on the website, but if you went to the very bottom of this website, there's a small link that says Publications and Manuals. And if you follow that and go to the Manuals section, you'd find the Researcher's Manual. And this Researcher's Manual, I think, is very helpful. I wish it was more prominently displayed on the website. It has a lot of great information to help you prepare your application. And in terms of questions that we field all the time, I want to make a slight change to my protocol. What do I need to do in order? A lot of that is already discussed in the Researcher's Manual, but if you don't want to read all the pages of the manual, you're also welcome to just reach out to us from the get-go. So here is the list of information that we ask for when you apply for a Schedule I research license. Whether you're a new registrant or if you're adding a new protocol to your already existing DEA registration, if you're adding new drug codes, or if you're using existing drug codes you've already been approved for, but you're doing new research with those same drug codes, some sort of supplemental or an amendment to your existing protocol. All of those would come in to us through an online system where we ask you for the obvious profile information. What is your name, your institution? You provide a CV as information for us to provide to the FDA for their review of the qualifications of the researcher. In terms of the research project, we obviously need to know the title, and we need a protocol, or at least a protocol summary, where it provides the purpose, the statement of purpose. In terms of the Schedule I substances, the piece that's often left out is in terms of the applications that we receive, is the amount of the Schedule I substances that you would be needing for your research and the source where you plan to obtain it. So you would provide that along with your protocol. There's some information that we need about the security provisions. So you have a statement of security provisions for storing and dispensing the controlled substances. And then if you're involved in human subjects research or animal studies, you would need an IRB or an IACUC approval. If you're not doing studies of that type, then you would still need some sort of institutional approval. If you're in academia, that could be a department chair. If you're not, it could be some sort of administrator, just a letter on institutional letterhead saying the institution is aware of this research involving Schedule I substances and this researcher wanting to do that research. And if you have an IND, then we would need to have that also. If you're obviously in the human subjects side, you'd be providing additional information related to the IND. So a lot of the information that we're asking for here is information that I know researchers have, even if they're not working with Schedule I substances. You would have a protocol. You would have a lot of this information. If you're not doing Schedule I research with humans, but you're still doing human subjects research, I know you have an IRB approval. Maybe the area that is a little bit different would be that statement of security provisions, where you would have, say, a safe that's secure and where you would store that those substances. Depends on the institution. Some institutions already have it set up in a way where it's dispensed from a pharmacy. Some individuals have it stored in their lab. However that is, that information is part of the application that we would need, and that might be a little different than some other types of studies that you might do. But the diversion investigator in the field is willing to answer a lot of those questions and help a researcher figure out the best way to implement that part of the application and the physical space that would need to be set up beforehand. And we can also help connect you with that diversion investigator if needed. So in that researcher's manual that I pointed you to earlier, there's this flow chart that shows you how these reviews are done. So a new registration, or even if you have an existing registration but you're submitting a new protocol or an amendment, it can be submitted through that online portal. It comes to us. DOE stands for our section. Diversion operations, and it's the evaluation section is the DOE. It's not Department of Energy, which is a little bit strange, I know. But the review process, so that comes to us, it enters our workflow. We make sure that all the information is collected. This is actually where there's perhaps the most delay is when we are waiting for researchers to respond to us because we're missing something. Typically, we would get the protocol, but we might be missing that justification for the total amount of substance needed, how many subjects, some of those details that we need in order for FDA to be able to do their review. But once we get all that information from the researcher, it takes us not much time at all to put that package together. It goes through a review process internally, just 2 levels of review to make sure it's complete. Not necessarily yet doing the scientific review. That's left for FDA. But then once we have that package ready, we send it to FDA. The field is notified that we have a complete package ready and sent to FDA. They, at that point, can go and set up their site visit, and they can be doing the site visit portion in line with the FDA review. Depending on the field division and the field office and their schedule, how quickly that happens is sort of varies in that regard. FDA is usually pretty quick about turning around reviews. As of late, I would say a couple weeks. Once we have a complete application and we put that package together and send it to them, it would take them a couple of weeks to get their review done. As soon as they send us back a letter telling us that it has been found adequate and complete, we again notify the field office to let them know that, and we enter it into our system as clearing our office. And at that point, if the field office has already done their part, then it would be an approved application. It would go to the registration section and it would be finalized and made official. If the field office hasn't yet done their site visit, then probably our relay to them that it's been completed by FDA and found adequate and complete would prompt them to go ahead and schedule that office or that site visit if they hadn't done so already. Some things that we've done to improve this over the recent years is that application process has been automated, or not automated, has been put online entirely. So there's no more paper applications coming in. We're not waiting for the mail to come in and to be logged, but rather it comes in to our workload online, and we're able to send out an email to get additional information from researchers very quickly. And so that process has really helped speed things up. We're also coordinating between our office and HQ and the field offices, trying to communicate frequently, trying to make sure that everyone knows where a specific protocol is in the process and what we can be doing at that moment to try and expedite everything as much as we can. We recognize that no 2 researchers or projects are the same, and so we try to work with everyone to make sure that we figure out what's the best way to get you to be able to do your research. And again, we are always available for questions and to help troubleshoot anything that you may encounter. Just a couple slides here of some quick statistics to give you a sense of how many protocols or applications we receive. So over the years, number of applications received, you can see it got up to, I think it was 252 in 2018. There was a bit of a drop in 2020 because of COVID, but those have started to come back. 2023 is low there because this was queried in June. And we hadn't yet received many of the applications that we would get over the summer months and especially beginning of an academic year. It's actually a very busy time right now, but so that bar there would be much higher now at this point. Current registrations include 842 active researchers. This I just queried this last week. In terms of marijuana and related substances, there are 494 active researchers. Psilocybin has 213 currently active, MDMA 170. You can see that over 40% of the Schedule I researchers of those 842 are approved to do research with some of the classic hallucinogens. We've seen a lot of studies recently involving psilocybin, involving MDMA, and I think this is obviously part of why you wanted to have this discussion today, and we're glad to have it. Many researchers also have multiple active protocols. So even though there's 842 researchers, some of them have 4, 5, 6 studies ongoing at any given moment. And so in terms of number of active protocols, it would be much higher than that. So in summary, we're glad to answer questions, but we just wanted to convey the idea that Schedule 1 research is accessible, and we're here to help. It looks very similar to other research you might be doing. If you're doing human subjects research or animal studies and so on, you would have a lot of the same information already ready when you're ready to apply for that Schedule 1 research registration. So hopefully the application process, the gathering of all that information isn't too cumbersome, and you can just provide that to us. We're glad to do the review and figure out what is needed if anything is missing, and try to communicate that to you as quickly as possible and get it through the process as quickly as possible. We do continue to streamline our processes, and something that we recognize too is that in terms of research getting done, it's critical to have the funding. So not only these registrations that we're dealing with, but also the general funding within the field to make sure that research is supported and promoted. So we don't have much that we can do with that, but we recognize that that is something of concern. So here's that email address if you would like to reach out to us. It's Schedule 1 Research, DPE Schedule 1 Research. So it's a capital I there, not a number 1 in the middle of that email address. And I will stop sharing.
Thank you, Dr. Hewitt, for your overview and for providing that contact information. I think that's going to be really beneficial for all of our attendees today. Apologies if you do get an influx of questions, but I think that's probably what the goal is, right?
Absolutely, and we have a team of people to respond, so that's great.
Perfect, perfect. So we're going to transition now. I know there's lots of questions building. I did want to remind folks to please put your questions in the Q&A box at the bottom of the screen, and we will get to those in just a few minutes. But before we do, I wanted to go ahead and take this opportunity to introduce the panel of experts who will helping be addressing these questions and navigating the questions with us today. So Dr. Yehuda, I will turn it over to you to introduce yourself.
Hi, good afternoon, everyone. My name is Rachel Yehuda, and I'm a fellow at the ACNP. That's probably the first reason I'm here. I'm also director of a Center for Psychedelic Psychotherapy and Trauma Research at the Icahn School of Medicine at Mount Sinai, along with the Bronx Veterans Affairs Hospital. We are running clinical trials and doing basic science work. We have 2 trials up and running right now, an MDMA-assisted psychotherapy trial in combat veterans at the VA and a psilocybin trial for PTSD at Mount Sinai. Thank you for having me.
Thank you for joining us. Dr. Koenig?
Hi, I'm Aaron Koenig. I'm the Chief Medical Officer at Psychedelics Therapeutics. We're a private biotech company located in the greater Boston area, and we're founded out of the academic work of David Olson at UC Davis. We're developing novel compounds that are inspired by traditional psychedelics that retain neuroplasticity promoting benefits while removing hallucinogenic as well as potentially cardiotoxic elements. And so very interested in this whole discussion. I also want to thank Tristan for sharing his story and Dr. Hewitt for the very nice overview.
Thank you. And now to the co-chairs of the National Network of Depression Centers, Psychedelics and Related Compounds Task Force, Dr. Fred Barrett and Dr. Tricia Supes. Can you please introduce yourselves?
Yeah, thank you very much. My name is Fred Barrett. I'm the director of the Johns Hopkins Center for Psychedelic and Consciousness Research. We're conducting a series of clinical trials in a variety of patient populations, primarily focused at the moment on the use of psilocybin for therapeutic purposes, also looking into basic human mechanistic studies. And I'm grateful to be part of this conversation. I'd like to hand it over to my co-chair, Dr. Supes.
Thanks, Fred, and thank you, Tristan, for sharing your story. I think it's very important that these stories are kept alive for everyone. Thank you. My name is Tricia Supes, and yes, Fred and I co-chair NNDC Psychedelic Task Force. I'm a professor at Stanford, also at the Palo Alto VA. We've just finished a psilocybin study for treatment-resistant depression in veterans really who have chronic severe depression. The majority of them also have PTSD. We're bringing a trial up now, getting pretty close to starting to screen for MDMA, assisted therapy with MDMA versus cognitive processing therapy, which is a VA gold standard treatment. And we're doing that also with implementation, feasibility, and some cost analysis. Also, we've been able to form a very nice network of about 6 VA sites that are all doing or will be doing either MDMA. Well, actually, this is sort of the MDMA group, but also other studies. Rachel is also part of that. So thank you.
And rounding out the panel today will be Dr. Resnick, who you met before, who introduced himself, but also brings a particular perspective to this discussions. Mark, did you want to address that?
Yeah, just in terms of what I do during the day, I study the neurobiology of depression, and we're very interested in biomarkers of depression and related aspects and how psychedelics have common cause with other antidepressant drugs.
Well, thank you so much to the panel for joining us today. We'll jump into the questions. I see some have been already entered into the chat, so please do keep them coming. And I'll ask Chantel to lead us off.
Thanks so much, Leslie, and thanks to everyone. I feel like in the interest of sort of bidirectional communication, I thought we would start off by having the panelists tell us from your perspectives, what are some of the barriers that researchers face to either starting or expanding their research programs into psychedelics specifically, in light of everything that Dr. Hewitt just gave us? Rachel, do you want to start?
I was hoping you would call on someone else first, but yes, I'd love to.
You were close in my view.
There you go. I found it difficult to get started, but I think that we also started about 3 years ago during COVID, when psychedelics weren't the biggest priority at the FDA or maybe even at the DEA. And so it did not take a short amount of time to get the approvals. I think things are better now. But the challenges are not only in terms of the government regulation. There are little challenges along the way with everyone, with just how long it takes even for the sponsor or for the institutions or for the IRBs, you know, so this is a little extra to what people would normally do. But what people normally do, you know, you don't realize how many hoops you jump through to do research. I mean, the average person does go through a lot of committees, maybe multiple institutions if you're at the VA or something like that. So if you just add, you know, 10 or 15% burden to 10 steps, it feels like a lot. But the perspective that I would offer is that it is possible to do this work. You just need to have certain things in place. You do need a safe that the DEA will approve. Most people don't have one, but you can buy one. I mean, so it's just a matter of, you know, a lot of little steps that get you to the finish line. Once you begin doing this research, it's pretty rewarding in that, unlike the kind of clinical trials that we've been involved in the past, you get a lot of interest in terms of people wanting to participate. So that makes you feel good that at least you're trying to do some work that stakeholders are very interested in having you pursue. So at the end of the day, I think it's a longer trip than you think, but there is an end to the journey.
Okay, I would just add to that doing it within the VA, in the first study, we had to bring everything up from scratch. The pharmacy at our VA would not handle Schedule I. So I really felt like I spent about a year doing problem solving and building things up from scratch. So it was about 3+ years. By contrast, our second, and yet once we started the study, we could have finished it in a third or done 3 times as many patients because there was so much interest, which I think is important. And we were able to help some people very substantially. This study, we're spooling up. It's going to be under 12 months from beginning of submission up the regulatory climb, including IRBs and so on, I think before we're screening. So I do think it's sped up, and the DEA per se is not a rate limiting step. It's sort of just the number of things you have to do.
I wanted to add a perspective from a basic researcher who may need to use many different compounds and may need to move quickly because you make decisions quickly and want to do an experiment the next week. You can't do that under Schedule 1. So I'm hoping that we can figure out some way to carve out research. Also, the safe, well, bolting in a safe to the floor and maybe providing electricity for it at a university is going to cost more than $10,000 because you have to use the university scheduled physical plant, and they are much more expensive than regular market. So you've got the safe as a problem, and you've got the inability to move quickly. And I'm talking about nanogram amounts of compounds. So really, something that is not that wouldn't be an issue for the possibility of it getting out. Although it doesn't get out. You still have a locked cabinet if you're scheduled 2 through 5. You still have a logbook.
Well, one thing about the safe issue is when we got our safe, it weighs 1,800 pounds. It looks like a bank vault, gave me greater appreciation for Netflix on people who break into banks. But when you open it, there's 4 compartments because it's my understanding, and maybe Dr. Hewitt can add light to this, that for each study, you need it. So it's a huge door, and then there are 4 lock compartments in it. So for each study, even if you're using the same compound, you need separate drawers that also lock. So yes, and we had to go to a special room to get the thing delivered, and it's not moving. And if there's an earthquake, I'm getting under the door because the building could come down and you might be just fine.
It's definitely a consideration where you would store that really heavy safe.
I just want to say that I had the benefit, and I still do have the benefit, of our research center existing within the Behavioral Pharmacology Research Unit at Johns Hopkins, which has been doing research with Schedule 1 compounds for decades. We have a dedicated research pharmacy, a registered pharmacy that does nothing but serve our studies across drug classes within the building. And even with our experience over the past 25 years conducting studies with psilocybin in humans and with other compounds, it still takes us about 9 to 12 months on average to get from first submission to enrollment. And so Tricia, you're in the zone. That's great. It boggles my mind having to come into this completely cold, the number of details. But we have an awful lot of resource now built up to support our multiple. I think we have 8 or 9 clinical trials that are either running or are about to kick off. So this absolutely can be done. It doesn't require a research pharmacy, but the barriers exist on many levels, and I'd have to say that the federal DEA is gotten incredibly easy to work with. Sometimes the field offices are not quite as efficient as the federal office, and then dealing with your local, your state controlled substances organization, whichever that may be within your state, can be a rate limiting step as well. It's remarkable how many hoops you have to jump. I'm going to reiterate that. Yeah.
If I could just add, I think this is why so many people create centers instead of just do one study. That the amount of effort that it takes to launch one study is very big. But once you've done that, it seems like you might as well just keep going because, this is, I would discourage people who just want to try this on the side and really have more main stuff that they're interested in, because this might actually be the tail that wags the dog. In terms of effort and energy. So But if you are interested in kind of redirecting your studies to do more of this work, then it's hard in the beginning and then you build on it and it gets a little easier. But I think the message I want to send is that it is doable. It's just harder than what you've been doing. And there are more steps, but we do hard things and then they become the new baseline. So I think I really don't want to be discouraging, but I also want to be very transparent.
Yeah, I'll just add I understand we're on an industry perspective here, but we just built out a brand new space and found it to be a very seamless process. I mean, we actually thought it was even a bit easier than we expected it to be, and we put in extra safeguards. So I wouldn't want for people to feel completely dispirited. It is very doable. In fact, we found the DEA to be very a great partner to work with here.
Yeah, I want to thank Rachel for really making that clear remark, because if I can do it, anybody can. You just got to start walking down the road and you just keep going till you get there and then you do it. And I think that we know so little really about the medical application of these compounds. It's really critical that even more people get into the field, and hopefully with somewhat larger samples. So yeah, thank you, Rachel. I think it's important. You got to be persevering. So
that's why I'm trying to argue for some kind of a carve out for research to let us for psychedelics outside of Schedule 1. Let's get more people involved.
Well, thank you guys all for weighing in on that. I wanted to get to a couple of the questions that I think are specific to Dr. Hewitt. So one question that came in was whether or not Schedule 1 licenses are specific to the researcher, the substance, or a specific protocol. So can you talk a little bit about whether or not somebody has to do an entire new protocol or new application, depending on whether they're using a new substance or not?
It's actually specific to the researcher at a location. So one researcher in one location has a DEA registration. If they want to amend an existing protocol to add new drug codes, they would do that under that same registration. If they want to add a new protocol, doing a different study, either with drug codes they've already been approved to use or new drug codes that they haven't used before, they would do that under that same registration. If they wanted to take that research and do it in a different location, say a clinic across town, then they would need a new registration at that location. And I know, Teresa, you didn't get a chance to introduce yourself, but do you want to say anything else about that? I don't know if you've been following some of the questions as well that might tie into this.
No, I think you're on point. Just a brief introduction about myself. I'm Teresa Carbonero. I've done research with psychedelics, preclinical to clinical, including my postdoc with Roland Griffiths, overlap with Fred for a few years there before transitioning to DEA. Hi, Fred. I've been a DEA for about 5 years with the Schedule 1 Researcher program as part of my duties, and I've probably processed a lot of applications coming in, and I've definitely seen the shift from like marijuana to a lot more psilocybin MDMA studies. So yeah, happy to be a part of the panel and help Will with whatever questions you guys may have.
And also, Dr. Hewitt and Dr. Carbonero, is it true you need separate storage for each of the protocols under a given Schedule 1? Is that correct?
Not that I know of.
You can put them in the same drawer. Okay.
I would present that question to the DI, and in the specific setting of that lab and that research that's ongoing. I'm definitely not an expert in terms of the storage requirements. That's why I said that's reviewed by the field offices and the DIs. But there are some situations where it requires a site visit, some situations where it doesn't require a site visit, because I think they know the structure that a facility has already, and they know that it would already meet the needs.
Thanks, appreciate it.
There are also state regulations in terms of storage, and that may vary for the different states and different things.
Yeah, and when you're all listing all the steps you have to go through to do this research, there was IRB and there were IND that I was thinking in the back of my mind, nobody mentioned the state part as well. There's that entity as well that you need to go through. to make sure you're meeting those regulations.
Great. There's also another question, and this is sort of along the same lines, as whether or not the DEA approves the vendor for a drug substance or product, and whether you approve whether it's consistent with GMP standards.
Lisa, do you want to talk about that one? I can certainly answer it.
No. Yeah, so we don't verify those GMP standards. It does have to come from a DEA registrant, so it has to be a DEA registrant that you get the Schedule I substance from. But you can, as long as it's a registrant, you can use whatever substance it is. And not every study is for clinical studies, so you may not need a GMP certified substance for in vitro studies or for the different process, or to synthesize a drug, it wouldn't need to be GMP verified.
And a researcher as coincidental activity can also produce their own. They can manufacture their own drug if they're needing to do it on site, if they wanted to do it within their own lab. They can also, if there's not a DEA registrant who has the product that they need, they can, again, through coincidental activities, they can import it from elsewhere.
Dr. Koenig, I think you had a question that you wanted to ask.
Yeah, I had a question. I didn't want to sort of interrupt. These are really wonderful, I think, process-oriented questions, and I think a very practical way of thinking about how to conduct research with these class of compounds. I was wondering if Dr. Hewitt or Dr. Carbonaro, you could speak more broadly around how the DEA is looking at differences between the individual compounds. I mean, I think we're painting it with very broad brushstrokes to say that they're all the same. And I'm also looking at some of the comments in the Q&A about the real addiction potential of some of these drugs, and whether they're sort of all being lumped together. I don't know if that's something you guys could kind of comment, or if we could discuss.
Sure. So our group only works with the Schedule I substances, and they are all grouped in the same regard in that way. A protocol involving Schedule I substances and Schedules 2 through 5 substances would be reviewed by both the schedule for a Schedule I license for those particular substances grouped in that way, versus Schedules 2 through 5 would be another registration reviewed in a different way that we wouldn't necessarily be focusing in on, and working with the FDA to do that type of review that we would normally do. If that makes touches a little bit on it. I don't think it necessarily answers your question entirely.
Well, and I think that part of the question is about moving the field forward, right, and sort of understanding the true profile of these compounds, and not having them be overly stigmatized, whether it's the traditional psychedelics or novel compounds that are based on them. And I think that's something that we're interested in from a company perspective, but I think some of the researchers in the Q&A are interested in as well. It's about how do we sort of evolve from where we are today. But I understand you guys are very process oriented in terms of your subdivision. So.
And I mentioned at the beginning of my talk that research is informing that the policy decisions that are being made and the regulations. And that research is important. I do wish that FDA was here. They could talk about their review of when a drug is deemed to have a medical use within the United States, and the process for doing that review and moving it, say, from Schedule I to be considered for other schedules, or to be descheduled, whatever it may be. There is a process for that, and that is very much informed by the research that has been done. So in terms of continuing that research and moving those initiatives forward, that is part of that process.
There are a couple of questions that sort of revolve around the same thing. And I think you covered it in your slides, but maybe you can just state one more time sort of where you are in terms of reviewing the actual protocol, as opposed to what your role is versus the FDA.
Sure. We are a group of scientific staff that we're not doing the review for the merit of the research. We are really just making sure that all of the information is gathered. We prepare that package for FDA. They do the review, the formal review of the research in terms of the merit of the research and the qualifications of the researcher and so on. And that's housed at FDA, controlled substance staff.
Thank you. That's helpful. Are there other opportunities for the community, the research community, to interact with DEA to bring up stakeholder concerns?
I think that's what this is, right?
Outside of this forum.
Yes. That's that was one of our goals, right? We want to do this outreach. We want to hear what everyone has to say. If there's a way that we can help improve our processes, then we want to do that. If there's an opportunity where FDA can also be represented with us, that would be welcome as well. We want to have meetings, and we do have meetings with FDA on a number of topics because we interact in so many ways with what we do, just even outside of Schedule I research, in terms of that communication with them and doing the work both of DEA and FDA in tandem. So in that, gathering that information from the public is very important to us. There's a big gathering at DEA tomorrow and the next day. They're doing gathering information from the public. So that certainly does inform what we do at DEA, and these interactions are certainly important to us.
We've had a number of questions, as you can probably imagine, talking about what the process looks like for changing the scheduling. Whether that's just changing the scheduling for research purposes, or if it's changing the scheduling in light of evidence that comes down or the approval process. Can you tell us a little bit more about how that process is initiated, and whether you think, it's an automatic process? What does that process entail?
Teresa, I don't want to hog the stage. Do you want to say anything about this one?
Yeah, so first it's going to have to come from FDA, HHS for the drug to be recontrolled, and really until that happens, it will react adequately. There's not really much we can say just because we don't have any evidence. It's not medically approved. So until that happens, there's nothing that we can do to change the process. That would really, again, if FDA was here, that would really be a better question for them than for us.
Okay.
But one of the things we're also asking is changing it for research, if possible. Is there any way to do a carve-out?
So I'm not sure what that means in terms of a carve-out for that is what the Schedule I research program is to be able to do the research. I guess I'm not following.
I guess I was talking about the small amounts that are needed for laboratory research, for basic lab research rather than clinical studies.
Yeah, there were a couple of questions related to sort of the small amounts that are needed for preclinical research and whether or not that kind of changes the process for approval, since the amounts are so diluted out.
In terms of our review, it does not necessarily change. If I'm thinking of all the steps that everyone was mentioning at the beginning, you are going to be able to bypass a lot of those steps. For example, you don't need an IND. You might not be going to an IRB or an IACUC. You would be already sort of, for the basic research, you would be a much more direct path to being able to do that than in going through all of those time-consuming steps. If you are doing the basic research, say assay development, and you submit a protocol to us, those protocols are not nearly as long and exhaustive as the human subjects research. They are very much like a summary of this is what I plan to do with these substances. It's a justification of that. And it's pretty easy for us to make sure everything is there, for DEA or for FDA to do their review of that. And those do seem to sail through pretty quickly. So I believe that that type of research does get through the process just because more quickly, just because of the type of research that it is.
So we're coming up to the end of the hour, and I thought it would be prudent to end with a question to you, Tristan, if you're still there. Why do you think that the veterans population is really leading the charge and ensuring that psychedelic research and these treatments are given the attention that is needed at this time? And what do you hope that agencies such as DEA can learn from stories such as yours?
Well, I mean, so why do I think that veterans are leading the charge? I think if you're looking for a population that really could use, that are suffering from sort of a spectrum of injuries, whether they be moral injuries or physical injuries, or post-traumatic injuries. I think if you had to handpick one population that's sort of where these types of injuries are concentrated, I think that's the population now after 20 years of GWAT. I think that's a really like logical explanation for that. And so obviously when you have a high concentration of people that could benefit from something, whether it be illegal or legal, you're obviously going to have probably a higher level of those who are advocating for it. And what was the second part of your question?
What do you hope that agencies like the DEA can learn from stories such as yours?
Yeah, I mean, I think one of the things I wanted to emphasize is that, it wasn't something that I was seeking out. It was something that somebody else identified. Someone else had gone through, a similar treatment and identified that I might benefit from it. And so I was very reluctant, to try it, just because it's not part of my, it's not part of my particular like brand or culture or whatever. But that, once I sort of, drop these barriers of resistance to it, it was very incredibly beneficial. And so I hope that, organizations like the DEA and the FDA can look at veterans and see that, we're not, we're kind of, we look and speak and are as professional as everybody else who maybe had been advocates in the past, but maybe didn't have the personal lived experience to kind of bring with them in that advocacy. And so we're in your communities, we're leaders, we coach Little League, we work for Fortune 100s, we have kids, we buy houses, we improve, we're generally like baked in and try to improve our communities, and this is just that next step in trying to do so. So I hope that my hope is that you kind of see the authenticity in our advocacy and take that into account, because we're the ones that could use this the most. I would say at least one of the groups of people that could use it the most.
And also, Tristan, veterans are trained to lead the charge. So
that's it.
With that, we've come to the end of our discussion, and I really would like to thank all of the people who have attended and participated in this. I'd like to thank our panelists. I'd like to thank you, Tristan, for telling your story, and to all of you, thanks for listening, and thanks for your future actions. We look forward to further discussions and to charting a path forward. Thank you all for being here. Bye.